My background is generally as a mathematical modeler for biological or medical systems. So my background is not sort of fluid dynamics. The fluid dynamics interest has sort of come in having a look at the problem of bacteria biofilms. And in fact, it's not actually the fluid dynamics as my particular interest. The fluid dynamics was to try and get an approximation of growth as a backdrop to what I was interested in, which is the cell signaling mechanisms, which I'll talk very briefly, but I know most interest will be on the fluids. So bacteria biofilms. So these are colonies of bacteria in a static state growing on some sort of surface, normally with a fluid around it. So these are three pictures of the same bacteria in very different states. So here we've got three blobs, which are three individual bacteria called Pseudomonas aeruginosa. There was a brief mention of cystic fibrosis the other week. This is one of the major concerns regarding cystic fibrosis, this particular bacterium. But it lives in various states, and this particular one pictured here is a swimming state. So you've got basically the sort of rod-shaped bacterium, and hopefully you can see at the back, kind of a filament sticking out the back. That is a flagellum, and basically it spins round one way to drive it forward, then spins it round the other way to basically disrupt, basically to stop it. There's something called tumbling, which enables it to move in a different direction. So that's a project in itself of considerable interest and in complex fluids, and has been looked at, flagellar growth. But I'm not looking at that. I'm looking at this state. So swimming around is highly energetic. Standing still is considerably less energetic, and that's a considerable benefit to a bacterium. That if you find a nice place to live, why not settle there? And that's kind of the fairy tale version of bacteria settling on a surface and starting to form a colony. And this picture here, all these pink rods are individual bacterium. So you can see there's quite a few of them, and this is quite an early stage biofilm. So just to get an idea of size, well, you've got some idea. Each of these rods are approximately 2 micrometers long, and the width is about 1 micrometer long. But they do grow, split into two, grow and split into two. So there is variation. If you let this grow a little bit longer, so for example, as indicated here, then you've got what is basically a massive biofilm. So this is a tube, which is probably roughly 1 centimeter in diameter. And but for a small bore, which I'm not too sure if that's just been poked or what, but basically this whole tube has been clogged up by a bacteria biofilm. And it's quite pure, so there's not much contaminants in there, so that's probably maybe just 1 or 2 species, and they have formed a colony which is, a thickness which is 1,000 times, or more than that, bigger than a single individual. So that is a biofilm, and that's what I'll be talking about. So my name's John Ward, and so I started this work in the late 1990s, and as I said, it was to do with cell signaling of bacterium. And Liz Sockett and Paul Williams are the experts, so they're the ones who sort of made me aware of this situation. I worked with a suite of mathematicians, John King, who most of you probably heard of, but also Adrian Cobo as a postdoc, and same as Keith Andrews. And Mark Newstead was a student of mine. And also I mentioned to Julie Croft, who was a technician who worked with me. I will show briefly some experiments that we've done, and she was very much involved in that. Right, okay, so just a general view of the biofilms, and then sort of slowly moving into why fluid dynamics is a concern here. So biofilms are basically slimy colonies that grow usually on solid fluid surfaces. All of us, having been students in our life, clearly had times where the sink hasn't been cleaned properly, and when you stick your finger around a sink, you get this sort of brown, gooey, gungy, smudgy thing. That is a biofilm. Anybody who hasn't seen one is not telling the truth, certainly as students. So in Pseudomonas aeruginosa anyway, there is a kind of distinct stages of growth. So they swim around, they find an environment which is nice, they find a surface, and they somehow adhere to it. Once they've adhered to it, they start growing and dividing, and in time they start producing a substance known as EPS, I'll talk a little bit about in a moment, which expands the biofilm enormously. And what you get in the mature biofilm is basically not much bacterium. About 95% of that biofilm in the tube is water, in terms of sort of mass or volume, whichever you're going to choose. About 2 or 3% is this EPS stuff, which is a strangly polysaccharide type thing, and the other 2 to 3% is bacteria, live and dead. So, but there is advantages in living in this environment over this. This is a thin biofilm, up to about 20 micrometers. Any nasty agent can penetrate that easily. So if you've got an antibiotic, stick it up there, it'll diffuse down, kill everything. And it's also very easy to wipe off using shear flow and that sort of thing. However, if they're in this state, the EPS provides a considerable further rigidity, so there's much more resistance to shear. In fact, these bacteria live in rivers and things, so it wouldn't be much of a bacterium if you couldn't do that. They also, nasty agents have to get from here into there to kill the biofilm completely. So as the fusion limitations, and basically they are, as a consequence, difficult to remove if you want to remove them. So this is a young biofilm, or an early biofilm, and this is what I'm going to call a mature biofilm. And if you take a closer look at a mature biofilm, you get sort of live, dead cells, these strangly things as the EPS, and the rest is water. So that's kind of what's going on. So what holds the biofilm together? So I talked about that they swim around and they may attach to the surface. So bacteria are very small, so they are influenced by electrostatic forces, and some claim Van der Waals forces as well. And they believe that as the bacteria hits them, as long as they don't bounce off, they may be trapped in some sort of field, which enables them to stay there. They produce these things called fibri, which is these sort of proteinous substances which also allow adherence to a surface. So you can sort of see that sort of an increased surface area. So if it is electrostatic forces, then that's an increased surface area. These fibri also connect to other bacterium, but the range is around about 2 to 3 micrometers. So the bacterium has to be fairly close, sort of like a length scale of a bacterium. So in the early state, there is some mechanism by which they attach to each other, but it's not a strong mechanism and it's easily broken. However, in the mature phase, I am told, I didn't find too many pictures on the web that really sort of showed what I really wanted to show, but I am told that this is genuine polysaccharides here, which is EPS. So EPS stands for exopolysaccharides or extracellular polymeric substances. It depends who you talk to, but at least they're in agreement with EPS. So fortunately, it's the same appropriate letters. So the EPS are these strands here connecting to these blobs. These blobs are individual bacteria. So, so they form sort of like a hair-like structure, which if you put it under sort of stresses, they're quite viscoelastic nature, but I'm not going to be in a sort of a high stress scenario in the models I'm going to be looking at. It'll be very much low stress. So this is what was claimed to be not fibri but EPS connected to bacteria. The bacteria actually here are quite dense. I'm sure much more mature biofilms are much more diffuse than that. But it depends on what bacteria you're talking about. So why are we interested in biofilms? So I don't want to go into too much detail. So basically, we owe our existence to bacteria. They produce oxygen generation. They also owe our existence because they fix nitrogen into in soils. They help in our digestive process, and any fans of flatulence can thank bacteria in their gut. So they're totally behind that. The fact we drink nice clean water is because of bacteria biofilms, and various chemicals are produced by bacteria in bacteria grown in an industrial scale in what they call bioreactors. But of course there are nasty things about them. They cause infections, you get a wound, they infect and nasty things can happen. If you get a medical implant, they can infect implants. So there's an enormous amount of research into designing materials that are antibiotic naturally, not so the colonies can't start up there. Of course, contamination of water and food, poor prognosis in various diseases, cystic fibrosis is one. We've all got dental plaque. We get that, clean our teeth once or twice a day, so somehow control that. And of course, in industrial situations, they clog tubes and damage ship hulls. So basically, some understanding and therefore control of these things are important in all sorts of settings. So this is just a little bit about where my interest came in. So I mentioned this sort of magic cell signaling process. In fact, it's what's now known to be called pretty much widespread everywhere now. It's quorum sensing. So quorum sensing is a cell signaling mechanism by which bacteria can sense the presence of other bacteria and change their responses accordingly. And in Pseudomonas aeruginosa, this particular bacteria, when it's in this dense microcolony stage, so they have the fimbriae but perhaps no EPS, if you had the quorum sensing system working, they go and mature and produce all the big biofilm, which are difficult to remove. However, without quorum sensing, they don't tend to mature, and they tend to stay at this vulnerable dense microcolony stage. So the background to the research is to understand quorum sensing a lot better and perhaps have a means of targeting this process to control biofilm growth and various other aspects of bacteria behavior. So I'll talk briefly about that, but I will concentrate on the fluids a bit more. So here's an overview of the talk. So it's I've done so that these are sort of three aspects which I have done. I've done various other things as well, which no time to go into. But the first thing I'm going to do is just a simple model of thin film of the early biofilm growth. Then I'm going to sort of look at the more mature phase for a one-dimensional case and a multi-dimensional case. I think in a sort of a situation like this, being a school for people who are generally researching into fluids, the interest will be more perhaps on the modelling than the things I'm going to point out as the results. But so I'll probably labour on the modelling a bit more than I would normally do. So it's going to be three parts. I'm hoping I'm going to have time to talk all about them. So the first thing is going to be the thin film modelling. So starting from small colony to a dense colony. So the aim here was to derive a model of early biofilm growth based on the principles of classical mechanics. Actually, there was a model before the one I'm about to describe that we did use, which sort of basically answered our questions on quorum sensing, which did a few little cheats in terms of the classical mechanics. So he wanted to write a new model which cheats less, although there's a little bit of cheating, as all modelling is. So this is the model schematic. So this is what we've got. We've got a fluid media, and we've got our bacterial microcolony that are suddenly smeared out into a continuum. And not only is it a continuum, we're going to assume it's homogeneous. Which is, homogeneous is probably a fair enough assumption. Of course, the bacteria could discrete particles, but by instinct I tend to sort of look for a continuum limits to this, and probably stretch continuum limits a little bit on this case, but I'm going to assume that the bacteria form a continuum and it's of constant density. So the variables we're going to be concerned with is essentially the height of this biofilm and how that moves in time. Just by looking at observations, a microcolony without going maturity only grows up to about 20 micrometers, so very thin, and the width can be any distance you like. So the aspect ratio is going to be very small, HB over LB. So HB is the height scale, LB is the length scale, which can be almost anything you want, but we're going to say it's a lot less than one. As I said, the biofilm is homogeneous, and we're going to assume it behaves like a viscous fluid. So I said it's going to be a blob of stuff which is under very low stress. You can think of things like as bacteria grow and move around, they are rubbing against each other, they're rubbing against each fimbriae. So there's a kind of a very, very sort of slow-acting frictional force. And if you take the massive leap of logic that, well, let's take the bacteria size to 0. Essentially, friction between blobs to 0 is essentially what viscosity is. So it is very much a continuum assumption which sort of has a sort of a solid background. Should also say that 20 micrometers represents maximum about 20 bacteria high. So we are taking the continuum, so we're probably really right on the edges of acceptability in the continuum limit. But you could also view that this is a model of 1,000 cases of biofilms and we're simulating the average. You could look at it that way. Okay, so we've got our biofilm. There's nutrients in the thing, and basically nutrients is what generates growth. So bacteria need nutrients to survive, and basically they use nutrients to grow, increase in volume, suck in water from its environment to create the new volume, and then divide. Because it's creating new volume, I propose that it's pushing its neighbours to accommodate the new volume, and those neighbours are moving, and they're moving at a certain speed, which means there's a velocity. And if there's a velocity, that means there's some sort of pressure gradient. So we're going to have velocity and pressure there as well. I'm only going to be looking at the bacterial phase. There is the water in between the bacteria, and that's getting sucked in and being used to convert into bacteria mass. However, it serves no other function, and it decouples from the system, so I'm going to forget water in this case. We do look at it later on, but in this particular model, no. Timescales of interest is of order 1, and making these assumptions, defining these things with twiddles as the various derivatives and grads and variables, we use a Stokes flow approximation, and we get these two equations. Continuity equation with a function f, it's not 0, which is why it's a bit rare, and we get the Stokes flow equation, which has this additional term, which is a little bit unfamiliar, except to those who've actually sat down and derived it and wondered where that's come from, but of course you don't care because normally div u is 0. However, it is there, and it's assigned as a curious value of third, but that's to maintain the definition pressure as being the mean normal stress of any arbitrary blob. I'm sure one or two think, looking at that saying, where's gravity? If gravity, this number, this Reynolds number approximation is assuming biofilms made of water. If it was water, then you certainly do need gravity in this system. But biofilms grow on the bottom, on the top, and you can't really tell the difference between them. If you grow bacteria on a petri dish, and biologists totally don't care, they just chuck them in their incubator, upside down, right side up, they're all the same. They don't gravity appears to make no difference. So based on that observation, I've done the experiments myself, it is true that I'm assuming gravity is a negligible effect. And talking about nutrients, just simple diffusion of nutrients coupled with some consumption. So I'm assuming that's in the quasi-steady, so over 20 micrometer scale. If you're talking glucose or any small carbon source, then that's a reasonable approximation given the timescale of that. Okay, so okay, so this is where sort of gambling that the movie's going to work. So are these assumptions any good? Now the particular thing about this movie is to demonstrate the idea of the velocity field. So we've got 4 bacterium here, and if I press play, you're going to see bacterium grow in every good Invasion from Mars 1950s B-movie thing where these things are growing so fast. So over, I don't actually know what sort of times course, but as this is playing, or actually I might control it, just stare at one bacterium and then just watch it move as the thing develops. And it does move around. So the growth in the middle of the thing is pushing the bacteria out. So describing bacterial movement as a velocity field seems to be very valid. And I guess, unless you've thought about this, that this is perhaps a little bit of a surprising thing, that bacteria are, okay, they're mostly made of water, but they're not water, but they flow. So, so growth and division causes a tide of expansion. I really absolutely no idea how they've done this thing, so I presume it's a bacteria colony on a slide with a cover slip in some sort of medium. But it has, certainly in the early stages, seems to be growing in a 2D plane. There's sort of an orientation there, so they're sort of pointing approximately north-southwards. But we can see as it moves around that things get distorted and they tend to lose their polarity, so this is why they sort of expand in a semicircular fashion. But of course there is a three-dimensional world, so I'm a little bit curious as to why, at least at the start, that they appear not to have invaded the third dimension. But as you let the thing grow, things get a bit fuzzy here, which I'm guessing is actually layers of bacteria sort of impeding with the light. So it is growing in a 3D fashion. So that's where the idea, or where you can actually picture where all this growth goes on. And there's also the assumption that there's no gaps and things. It's a sort of a uniform thing. I think we can see that's reasonably uniform. So Right, okay, so we got on our Stokes flow type equations. So we're going to take the thin film limit, exploiting the small aspect ratio. And I'm going to consider two cases. So one case is where the bacteria attachment to the surface is weak. So and the perhaps not necessarily realistic assumption is that we're going to take things to the extreme and we're going to assume a free slip condition on the surface. So that reflects zero interfacial stress, which perhaps, although you've got to be careful with the limit, might reflect what a weak attachment would give you. Perhaps the more realistic case is the no slip condition, so they cause quite a strong attachment. And so we're going to consider that case. I'm going to index these two cases with phi equals 0 and phi equals 1. Apparently it is a homotopy parameter. But basically it's just 0 to 1. It's not a physical parameter. It appears in these things. It's not in the physical way. They don't tend smoothly from one to the other in a realistic sense. So phi equals 0, free slip, 0 interfacial stress. Phi equals 1, no slip, and well, we're going to have to include surface tension forces. I'll describe why in a second. So we do the standard thing. We look in the limit of delta tending to 0. These are the appropriate balances on the parameters. We plug them in, and this is the leading order reduced system. So the two cases, as I say, phi equals 0 will give you the free slip case, and phi equals 1 gives you the no slip case. That's made explicit in that boundary condition here, which is the sort of the tangential stress balance on the surface. And so these boundary conditions. So basically, we introduce the nutrients at the top. Can't go through the bottom. The usual conditions at the bottom were no flux across the substratum. The only thing okay, there's the kinematic condition, the standard one. 0 tangential stress, and the normal stress is given by this thing, depending on the two cases. Now, there is the issue of so when I first was looking into this, this wasn't a particularly popular inclusion of mine, but it's something that had to be done with the zero stress case. So imagine you've got your biofilm that does something like that. If the cells here are not allowed to move, then any kind of motion, any creep is going to be slow compared to the amount of growth. And because the cells here get a lot of nutrients, if you didn't have anything regulating it, you get something like that. So you're not getting growth outweight, you're getting a kind of a spike up there. So it's a high, it's a high curvature objects would form, which you can't allow with the thin film assumption. So we regularized that. And the simplest thing we thought of is saying, well, somehow the bacteria rearrange themselves so that there's a resistance against high curvature structure. And of course, that's just another way of saying surface tension. So we've got a surface tension term here. So that's only for the case 1. You don't need it for the case 0. Okay, so the nutrient equation is the same for both situations. So just a quick charge through that. So, sorry. Yeah.
You said that you also take, or that the bacteria are dividing.
Mm-hmm.
So they generate mass. Do you take this into account? Okay, that's the.
Yeah, it's there. Yeah, and it's there in the continuity. So, right. So, so nutrient consumption is just a sort of diffusion and some consumption term. If you look in the books, there'll be kind of all sorts of nonlinear terms to describe that. So we've assumed that, but more nutrients tend to mean that more gets consumed. So as long as it's monotonic increasing, we assume the simplest thing which we can solve, and that's a simple enough solution. The birth rate, this FSC, more nutrients, the more growth there is. If there's not enough nutrients, cells die, you lose volume. So we assume there's some sort of similar, a linear relationship here. So if C equals 1, which is by our scaling, it means FC is 1. If C equals 0, it equals minus rho. So it's a linear term. Minus rho is the maximum death rate of a starved bacterium. So assuming a linear term, there is an important quantity that's going to pop out a number of times, and it's the integral of the consumption term over Z. And can't remember, it's a long time ago, I put a half in front of it. So, and for this particular thing, that's what the function looks like. And without loss of generality, you can choose beta here, the consumption parameter there, so that big F of 1 equals 0. So that's a steady state. Right, okay, so case 1, 0 stress. So, so we had our model, plug in phi equals 0, that's what you get with the boundary conditions. You integrate over Z to get your vertical velocity, your pressure, and what ultimately turns into the height of the biofilm. To close the system, you have to look at the correction term, but in Cartesian geometry, x and y being the tangential directions, this is the final system that results from this analysis. So you got the sort of standard transport equations, and then growth. And here you've got these things sort of coming from the momentum equation. So, so you get this horrible system. No reason why you can't solve it, but I wanted to sort of make an extra level of simplification. And that's we're going to assume it's 1D, which shrinks the system considerably to this thing. There's still a little bit ambiguity here, so you've got d by dx and d by dx there. So in order to integrate it and evaluate the constant, or function of time, we assume that they've got this set up. So the initial conditions are some even function, compactly supported, such that x equals 0 is the center and x equals s is the point or the edge. And as the biofilm is going to grow, this coordinate is going to move, so it's a moving boundary problem. And what I just said is summarized conditions. So, and we close the system off. You don't have to. If you assume that h equals 0 on the contact point, then you can integrate that and you get this reduced system, which is much easier to handle with. And more or less straight away, if you got up early in the morning, had your breakfast, you would spot that if you make this transformation, phi equals 1 over h, it actually reduces to a transport equation equaling 0, which means phi is a quantity that's going to be conserved, which implies that 1 over h integrated over the entire thing equals the equivalent from the initial conditions. And what that tells us straight away, that how the long time behavior is going to be determined by the slope as it tends to 0, such that if you assume it's some algebraic decay, 1 minus x, and a lies between 0 and 1, as x tends to minus 1, you can prove there's a steady state solutions existing for that. Because this term is finite, therefore that will be finite. However, if A is outside this range, if A is greater than 1, then in fact this quantity is infinite, so there is no steady state, so you're going to get growth. So that's a nice result we can get more or less straight away. But in fact we can actually make further progress by what the so-called hodograph solution. So essentially a hodograph solution is an application of method of characteristics and then doing a little bit of fiddling about with the order of differentiation. You can actually use this term to solve this fully. So if you just look at this equation here, so the first order PDE, so characteristics, you apply the method of characteristics. Everything's sort of known except for this U, and you can use this equation in a sort of kind of a characteristic world to close the system and eventually solve it. So what happens is, so you do your sort of characteristic change of variables. T is like the time variable, big X is the a parameter that defines which characteristic you're on. If you solve it, you get this implicit solution for the height, where this function here is the integral of 1 over f dh. So that's the sticking point of this, because that's not always an easy thing to integrate. And then you can close the system off by following this sort of like the hodograph process, from which you can get all your information, get x in terms of big X and everything like that. So it turns into solvable. It can be useful. Unfortunately, the form I showed you earlier for big F of H, you can't use this, you can't integrate that basically. But with qualitatively similar forms, you can do integrate and you get closed form solutions. So in particular, so you've got this equation for H, and you've got this for the contact point here. And in particular, as T tends to infinity, in this particular case, you get linear expansion. So we can solve it completely for this system. So just a few numerical simulations just to show what was going on. So somewhere here is the initial conditions, and in fact, if you look at the scale, it's growing outwards much more than it's growing upwards. So it's growing, expanding along the surface, and then growing upwards, sort of forming this kind of shape. It is dependent on the initial conditions what sort of shape you get, but that sort of thing. And here is the speed of the contact point, the contact line, which tends to some linear profile in this particular case. And something that's important just from a sort of biological perspective, where is all the action happening? Big F is the growth rate, and basically it's saying that in the region at the edge of the biofilm, so not in the middle, at the edge, is where you're getting most of your growth and where most of the action is happening. Okay, so so that gives a sort of so that's basically the sort of picture from the free slip condition. The probably more realistic case is the no slip one, where we've got strong attractive forces. So we're playing the same game here. So we've more or less seen a derivation of this already. So again, you integrate over Z, get your pressure in terms of the curvature, and you work out these things. Imposing the kinematic condition gives you this equation for H, which results in very nearly the standard thin film equation. So if that F is 0, then that's sort of the sort of more or less the simplest case of an equation, which scared me when I sort of first derived it, because I knew lots of people have done this work on this, and there's me deriving it, not knowing much about it at all. But what's rare, perhaps unique, on the study of such equation is having this source term F. So in this case, this is just because bacteria is generating volume. So I'm going to show some simulations just to sort of give an indication of what this will predict. It's a kind of a school. Oh, sorry, this is the problem. So the problem I'm going to sort of apply this model at is again a very similar setup. So we've also had all sorts of discussions on contact angles and that sort of thing. An alternative approach, rather than imposing an angle, is to impose a precursor film of some height, which is small, which I'm going to do that because it was basically numerical convenience at the time. So the biofilm is growing on a pre-wetted surface. This brown bit is where the bacteria are, the green bit is where there's no bacteria. So there's some kind of artificial blockage to preventing bacteria going through that. If you allow bacteria to go through that, then the whole solution structure will break down. So, so that means you had to have a slightly modified F, which is just given by this. So this is the slightly modified thin-film equation. And these are the boundary conditions, and I'm highlighting in red H equals epsilon, the thickness of the thing, which is important. So, so in order, before I show simulations of this, once again, I'm going to just a brief discussion of what the situation is at F equals 0. As it is a school, nobody's actually sort of mentioned the word, or not that I've seen, similarity solution. And I think with a problem like this, similarity solutions is important. So if you've got an equation like this, and you've got some initial condition which is compactly supported, and for convenience, it's an even function, then the large time solution follows a similarity solution, which can be determined by the following ansatz, which often Baron-Blatt is associated with them. So if you plug in h equals t to the power p g, and eta equals x to the tq, so you do a sort of a change of variables here, you plug this into that equation, it gives you a relationship between p and q. But there's also, because there's no creation or loss of mass here in the assumptions, we have to have mass conservation, and that gives a second condition on p and q. And if you do that, you find your values, and that gives you what the appropriate solution is in the limit of large time. So this is what the function g looks like. So it's just a boundary value problem. We can get three conditions. So it's a third order system, but we've got one unknown. It's just an eta, so it's a compactly supported solution on a domain between 0 and s eta, which is an unknown value. So you've got these three conditions which come from what we know already, but because of this s eta, that's not enough. And this is where all that discussion of the contact angle comes from, because we need to impose something else in order to close the system and solve it. But from my point of view, the most interesting thing is actually how fast it grows. So you can forget all about the contact angle. This is the bit that's interesting. So the contact outline grows like t to the power 1/7. So, and if you're working in radial symmetric geometry, it's t to the power 10, which I think was mentioned in another talk. So that's the sort of the backdrop. So without growth, that's what happens. With growth, this is what happens. So selected a value of epsilon equals thing, some value for h min. And what we get is these solutions. So this is a evolution in fixed time steps. So what we can see is a very near clear linear expansion of the domain occupied by the biofilm. And this is F, which, like before, shows that the growth happens at the edge of the biofilm. That's where all the action happens, and that's moving along. So if you plot the contact line against time, it tends to the velocity of the contact line, it tends to a fixed value. And this point S is just the coordinate of h min, isn't it? It's the value of x where h min equals 0.02. So, of course, it's arbitrary how you sort of select your point. But that's how I picked it. And the contact line, so the contact angle, h of x, goes to a steady state, and that's quite important in the analysis. Which, okay, interesting mathematically, but slightly disappointing as a modeler. So we got sort of constant, solutions expanding in a constant speed. So that motivates us to look at the travelling wave limit of the model. So if that's our equation, subject to those conditions, we rescale to a long time scale, or translate to a long time scale using z equals x minus st. So st is the coordinate. This big T is the time scale at which the travelling wave solutions develop. So it doesn't develop in order 1 time, it actually develops in log 1 over epsilon time. So if you substitute these things with a little bit of careful boundary layer theory, the equation that sort of wears the trousers and governing on what's going on is this ODE problem here, boundary value problem here. So you've got a fourth order ODE, and these are the conditions. This gamma, which is the derivative, so naught is the contact line point, and this gamma is the gradient of that, basically the contact angle of that point, and that's one of the things that you have to find as part of the solution. And it turns out that the growth rate is 3 times this value, which when you go back into normal time is something that's some constant divided by log 1 over epsilon, much to my disappointment. So, so it's dependent on so the speed is dependent on the precursor film thickness. The precursor film thickness is an artificial introduction. It was something I just imposed just to, you know, do the numerics. And it's a bit disappointing when something arbitrary actually is very important as part of the solution. So it means that there's something missing in our model, and basically it's a little bit of data is needed. So, and that's basically some knowledge of the contact angles or some knowledge of the physics of what's happening at the edge of the biofilm is absolutely imperative in order to really close the system. And just to demonstrate the dependence on the that precursor film thickness on expansion. So this is a plot of wave speed. Here is the nutrient concentration. More nutrients, more bacteria grow, so things grow faster. And it appears that as nutrients increase, the wave speed grows fairly linearly. And these are 3 different values of the contact line. So there is a dependence of speed on the arbitrary parameter. So there is a lot of work still to do on that, and basically hard physics needs to be done on that contact line. Right, okay, so that's the numeric story. I'm going to have a brief word about quorum sensing, because that's what really motivated the whole process. So quorum sensing, what is it? So basically it's the apparent regulation of behavior in response to population density. And a really nice example is what started this off, and it was when somebody who was growing bacteria in what they call a batch culture, basically a flask, and it stays dark, and then all of a sudden it lights up. These bacteria are bioluminescent, but not only that, they seem to switch on their bioluminescence genes you know, at a certain point, and they learned that it's at a certain population density, and as I say, it seems that their population almost spontaneously lights up at the same time. So this is actually a curve from a simulation, but I'll point to it just to give you sort of the idea. So if this was actually bioluminescence, and this is ours, so your population, this would be an exponentially growing population. So you've got no light here and lots of light there, and a relatively rapid transition from no light to the other light. And you can see probably a student playing games with a Petri dish and growing the species of bacteria, writing its name with a smiley face, showing that bioluminescence there. So it took them about 20 years to actually fully understand what the mechanisms was going on, and they identified the sort of sequence of genes which is controlling it. And when they published that, other people saw in their own particular bacteria, they had the same sequence of genes. And all of a sudden, quorum sensing was seen absolutely everywhere in every single species of bacterium. So E. coli is an example that gives you sort of a bad guts. Staphylococcus aureus, you've got tons of it on your skin. MRSA is the bacterium that is Staphylococcus aureus, who's got particular rightly bad press. Yersinia pestis causes Black Death. That's a pretty dramatic case. And in the interest of balance, not all bacteria are bad. This is a Lactobacillus casei. That's the bacteria that's in your a certain yogurt health drink. So they call them friendly bacteria. So, and the bacteria that I'm concerned, Pseudomonas aeruginosa, is involved in bioformation as well as a number of bad things. So if you ever had an infection which appears a bit green, that's almost certainly Pseudomonas aeruginosa, releasing a chemical called pyocyanin and pyoverdin, which are green, basically chemicals that, based on cyanide, that burns into your tissue. Not very nice, but that's also quorum sensing control, but I'm only caring about biofilms. So it's regulated by some diffusible signaling molecules, which I'm going to call quorum sensing molecules. And essentially, this is something that they produce. They detect how much is in the system, and if there's lots of it, they change their behavior. So if you ask a biologist, there you go. They'll give you something like this, and they ask you, can you model this? This is my gene. I really want to know what that does. So we've already got a biofilm model. I don't want to model all that sort of system here. I'm just going to model something that kind of reflects what's going on. The quorum sensing molecule concern is this thing here, so that chemical here, and there it is. That's the only thing I'm going to worry about on this particular pathway diagram. So, so the question is, which is what's right from the start, is what the key parameters required to activate quorum sensing and hence biofilm maturation. So at the time we had a sort of limited experimentation on this. Very, very difficult. Oh yes, when I started this, parameters for this, nobody knew any single parameter whatsoever. In fact, our group was probably the first in the world that worked out the decay rate in serum of this chemical. Biologists don't tend to understand dynamics very well and the importance of decay rates. But anyway, so we did a few simple experiments. So we got some data, but it wasn't brilliant data. So the model has to be simple and reflective of the data that we had. So we've got H and U. So those the biofilm model. That's already there. That's done. And for our new model, we just assume that we've tracked the concentration of the quorum sensing molecules, and that the population of cells are divided into two parts. One we call downregulated, which effectively don't produce any EPS, but also produce QSMs at some low rate. And then we've got the second set, the upregulated cells, which produces lots of QSMs and produces EPS. In this particular model, we're not going to look at EPS. All I'm concerned with is the production rate. And we said that all this is summarized in basically two simple reactions. So downregulated plus quorum sensing molecule makes upregulated, upregulated, spontaneous goes down downregulated. Few other sort of technical issues, like quorum sensing molecules over the timescales in a quasi-steady equilibrium. The equations here is actually what you get from reducing it to the film limit. So there is an escape constant Q, which is basically comes from a mixed boundary condition for saying the escape rate of quorum sensing molecules from the biofilm. But when you integrate things, it just you just get a simple term coming out of it. And there's some sort of technical details, negative regulation. So the equations that we got here, so we've got a transport equation for upregulated cells. So that's the mass action kinetics for this reaction here. Downregulated cells can be expressed in terms of H, constant, and upregulated cells, and then we've got some boundary conditions. So just chucking that on computer. So in this particular case, we got to the sort of as the biofilm goes, grows. That's the dashed line here. You're getting quorum sensing molecules growing up and increasing, and here is the amount of bacteria which are being upregulated. So these are the ones producing light, if you like. So at the start, biofilm's grown a bit, no light, but after a certain size, they're producing light. And this is the fraction of cells being upregulated. So at the start time, up to about time 5 or something, not much action, then lots of action by time equals 10. One of the things that the biologists didn't expect was that not all cells do get upregulated, not at least predicted by this model. You only get a certain fraction that get upregulated. And that's because the spontaneous downregulation and upregulation is happening quite fast. We looked at limiting case. You might have pointed out there's a small parameter there, which is not the same as the thing. So the small parameter, we did some analysis on the singular perturbation. And the thing to feed back to the biologist is, we've got this constant here in terms of parameters that we could at least measure in a batch culture. That is the parameter that governs whether you're going to get upregulation at all. So if this term is negative, you get no regulation. If it's positive, then you get upregulation, and you can work out timescales for when all this happens. And that's basically a picture summarizing that analysis. So, in what I'm about to show, there will be quorum sensing happening in the background. I'm not going to talk much about it, so that's probably not the specific things. But that just sort of gives you an idea that we do have a model there, and that's the kind of thing it does.
Sure. So that combined parameter, is that essentially balanced between the timescales and decay rates of the diffusion and upregulation?
Absolutely, yeah, absolutely. So this is kind of the rate of the forward reaction, ND plus A. Beta is the reverse reaction. Lambda is the decay rate of A, and Q is the escape rate from. So if you're thinking about designing a drug that controls this whole process, lambda is probably the easiest one. You can send in sequesters, as they call them, which soaks up the quantum sensing molecules, which could reduce this number and hence prevent that. So if you can put that in a spray and aerosol for people with cystic fibrosis, that may have useful thing. Oh, I should say, people in cystic fibrosis, when they're in a rather bad terminal state, and unfortunately they do have to sort of cough up huge amounts of phlegm, the biofilms are there have been described by Paul Williams' collaborator as made of rubber. So the modelling here certainly wouldn't be suitable for something like that, but it's not a nice thing to have in your lungs. That one?
The spatial structure between up and down regulated?
So for the down-regulated, you can approximately sort of minus that with that. So notice that a lot of the growth action is happening here. That was on an earlier slide, but in fact most of the quantum sensing action is happening in the middle. So there is quite an interesting balance going on between the spatial, or the activities of certain spatial in spatial distribution of activities in the system.
You've got a precursor film underlying all of this you've chosen.
Right, this particular simulation was for case 1, so this is the free slip condition, so there's no precursor here. But yeah, if there was precursor, then it would be the same things we observed before. Right, okay, so that's the thing I'm going to talk about in quantum sensing. So,
What about diffusion?
diffusion of... there's no diffusion of cells we're taking. I mean, like in that film.
Not diffusion, but sort of they wiggle around.
Right, okay. Right, there is a lot of people, there is a phenomenon known as twitching motility, which basically they fire out a pili and then they like harpoon something and then drag themselves along it, which gives them a mechanism to diffuse, but because they don't have a flagella when they're in biofilm. But when you saw that video, how the bacteria are all sort of packed up, I'm afraid to say there will be people who would model diffusion in that situation, but clearly there's no space for the bacteria to move other than being pushed. So I've assumed a neglected diffusion in this system. It may be, I neglected it in the model I'm about to say as well, but it may be more appropriate in that case. Okay, so looking at a sort of mature phase, so I'm going to look at a 1D model which highlights most of the issues that's concerned with this, and 1D is obviously simpler than looking at multi-dimension. So I've shown this picture earlier where you've got this sort of structure of the biofilm, live cells, dead cells, EPS and water. And we're going to assume that they occupy volume in space. So every bit of space is occupied by one of these 4 things, so their fractions will add up to 1. So, but we're going to sort of follow the actual fraction, spatial fraction, volume fraction of each of these elements. It's 1D, so we're just going to model the depth. So it's going to be Z equals 0, which is the substratum, Z equals H, which is the top of the biofilm where things come in. You've got birth and death processes. So like that video showed, if you create volume, you have to shift a cell in order to accommodate that volume, so that cell is moved, velocity field. And there's going to be, we assume that some volume is going to be lost when a cell dies. So the volume will come sort of a dead cell, and the remainder will just become water. Nutrients come from the top and they're diffused down and advect with the flow of water. And there's also got quorum sensing, but I'm not going to talk about that. So this is the sort of structure that we're envisaging. So we've got these 4 sort of key variables that describe the structure. Nutrients is going to direct growth. EPS is going to be governed by the quorum sensing, so that is contributing to growth. And and there's going to be a velocity field to describe the motion. So these are the variables. So I'm going to look at the fractions. So I'm not going to look at concentrations or densities of things. You have to be a little bit careful when you model those and handle things very carefully. So dealing with fractions is a very good idea if you're going to sort of undertake a model like this. We're going to assume that the live and dead cells in EPS are going to be moving with the same background velocity. But water is going to have its own velocity. So water can slip round as something moves up. So you're going to have a cellular velocity which describes the speed of, or the velocity of, live, dead, and EPS. The water velocity has its own, velocity, and h is our depth variable. Got nutrients diffusing around, and then we've got our quorum sensing, which is the same type of ideas, upregulated, downregulated quorum sensing molecules. All space is going to be occupied by one of these four states, so being fractions, they add up to 1. And the down and upregulated add up to the phi L, which is the live cell thing. Okay, so we write down equations to describe this. Now this is where, fortunately, you're going to have to put my mathematical biology hat on, and there will be parameters. So essentially, the left-hand side of all these things are the standard transport rates of the various cell types, and the right-hand side is the rate of change. So so here is basically birth minus death rates. So FB and FD are functions. We kind of resigned ourselves that this work is probably going to be more computational. So we've actually assumed more realistic functions. These are sort of the types of things you find in papers with parameter values for CD and whatever the equivalents there are. When a cell dies, part of it goes to water. Delta is that fraction. EPS is produced by at a background level and by the upregulated cells. A lot more by something. It depends on nutrients as well. But we assume that the rate of EPS production is the same as the birth rate. So. And there's some sort of decay of thing. And basically water is lost when a cell's born, as the cell increases its fraction, so as it grows bigger, it sucks in water. But when a cell dies, it releases some water, so there's a plus term there. Add these four equations, and you get sort of a kind of a two-velocity divergence equation. I never was a big fan of this, but Keith Angridge, who was doing the work here, was sort of insisted upon it. Essentially, EPS is made out of polysaccharides, so its manufacture is just by taking things that have no particular volume. However, they produce volume when, sort of rather magically, to produce the EPS. So in this situation, I prefer that to be 0, but he wanted to do it this way. So there's a non-zero divergence of velocities. The moving boundary moves at the local velocity. Nutrients, exactly the same before, diffuses through all the live cells, dead cells, and water. Assumed EPS is impenetrable, hence the appearance in the diffusion term. And then there's the terms for the quorum sensing. So if you add up all the equations, you'll find out that there's one missing, so we need a constitutive relation. And the constitutive relation here we chose is to say water equals some background plus something proportional to the amount of EPS. So this is based on the idea that EPS is basically loads of polysaccharides. They're connected with oxygen groups. And oxygen groups don't like meeting each other, so there's electrostatic repulsion of EPS. And if there's electrostatic repulsion, there has to be volume that fills the space to allow it to repulse. So we assume that water is moving in. Oops. So we make this assumption, which is fine in 1D. That closes everything up. So using that assumption, fiddling about, things sort of drop out a lot easier, and we have a slightly reduced model of what we're doing. And then we've got the simulations about to show. We've got some boundary and initial conditions. Anything to report? So you've got initial height. So we've assumed that all the cells are alive, no dead ones, no EPS. no upregulated cells or downregulated. Zero flux conditions at the base, and add nutrients, and we assume that quorum sensing molecules are lost completely on the edge. So this is a sort of type of thing, we did lots of investigations with this model, certainly not going to go through it all. But the first equation was the sort of kind of thing the biologists are interested. They could switch off genes, they could switch off the genes that produces EPS, or switch off the quorum sensing quorum sensing genes so they don't produce the EPS. And this is what the model predicts as the difference between a wild type and a quorum sensing negative thing, and probably better shown here, the growth rate. So wild types grow faster than the negative thing, and that's exactly what's seen in experiments. Quorum sensing behavior, so this is sort of the quorum sensing behavior, the jump type thing, this is a logged axis. If it's our bioluminescent bacteria, no light here, lots of light here, so they produce a light, and the negative ones obviously don't produce any light. So this is sort of kind of thing that was expected. As I say, we did all sorts of investigations with this. There's in the sort of a large time for a large biofilm, these are the distributions of various nutrients and whatever. We've got water velocities and cell velocities and the components. And by tuning the parameters a little bit, we can get the right sort of proportions of live cells and water and EPS. So some parameters we knew, diffusion rates are generally known, others we have to sort of guess and tune. Okay, Keith Angwich was very much an enthusiast of the comparison theorem, and he sort of found various ways to, even with a complicated model, to get some results known to be true, but it was nice to get some kind of proof. So this is one example, is that if we've seen that in this picture, we get a constant growth rate. He demonstrated, at least he's shown, that you can bind the growth rate between two constants through these relatively simple arguments, in fact, of getting upper and lower bounds on the variables, even though these look horrible, but you can say FB is less than 1 and FD is 0, whatever, and you can sort of get your upper and lower bounds. So there's a few analytical results, but analytical results are very difficult with a model of this type, and that's one of the downsides of a model which is perhaps the most biological interest of biologists. It gets a bit hard for us mathematicians. Okay, when got the student Mark Newstead, so this stuff was just about being complete. So the next sort of thing was, well, what about if you model this in 2 and 3D, or 2D specifically? Are we going to get any kind of interesting behaviors? So one thing that is not really known about is what are the flow regimes? What is water doing inside a biofilm? And that was one of the questions which sort of intrigued me at the time. And it also gives us an idea of how transporter molecules, particularly large ones, because there is an issue of transporting molecules. Can we exploit the flow regimes inside this biofilm to better transport antibiotics or anti-quorum sensing, whatever thing you want to do to control the biofilm? So the modeling is, we took the same sort of approach. We assumed that now we've got a sort of a 2D structure, so growth could be anything like that. In the 1D model, we had this constitutive equation, which is absolutely fine in terms of model closure in 1D. Unfortunately, it doesn't cut it for 2D. So we had to go a little bit to the drawing board and took inspiration from mixture theory. And in fact, well, there's more inspiration from a model for tumours, which was taken from mixture theory. So basically, the idea is we got a multi-phase thing. We've got alive cells, we've got dead cells, we've got water, we've got this EPS. And we've already decided that the non-water phases move at a certain velocity and the water phase uses other velocity. So there is going to be some sort of pressures and forces involved, and we wanted a way to capture that process. So again, we're assuming that there's no not much stress involved, there's no flow over it, it's just a static biofilm. So we took a viscous approach. So So basically, assuming that each of these components are viscous, or at least the solid phase ones, you get your sort of standard stress tensor for that. We've assumed in comparison to the various between the sort of the force between the solid phase, that the viscosity of the water is relatively negligible, and we just assume that the stress tensor for water is just simply pressure on the diagonal tensor. Now the force balances, which is basically where the idea of the mixture theory comes from, and it says basically you equate the forces on the solid, or the various phases. Here we've just got solid and water, and basically you've got your stress tensors for each of the components, and you multiply by the fraction. So this is kind of like a weighted mean of the solid stresses, and if you add to that some body forces, then the net force is going to be 0, otherwise you're going to get acceleration of the biofilm. And likewise we have a similar sort of thing for water. So basically within these sigmas and these f's are going to be described forces between each of the components and various other body things. And essentially we assumed we took a Darcy-Brinkman type model to describe the interaction. Darcy's law you've seen as basically, so Darcy's law is essentially something like the Brinkman part is adding viscosity, and unfortunately I'm going to get the sign wrong, so there's going to be some term like that. If you're looking at say flow over a porous media, so you've got the porous media equations and your porous medium, and you've got the flow, and there's sort of an interaction between the two. You can't close the problem properly in the porous media phase because of the boundary conditions won't match. And the only way you can regularize that, or a way of regularizing that I should say, is to add that viscous term in. So so essentially we've got that sort of idea. So that's the Brinkman bit, and this is the Darcy bit. Of course we've got velocities, two velocities here. We've got the velocity of the solid phase and the liquid phase. So V minus U is the relative velocity, which is the acting force of the drag. So you've got the drag terms. So we devise these so that the body forces all add up to 0, and this term here, which I have seen two talk by engineers who did not have this term when they were talking about mixture flows. This is quite important because I don't actually know, it's either dilution or dilation, I just get a bit confused. But if you did not have that term, what would happen, you're basically going to have a div p phi there. If p is constant, it implies that you're going to generate a flow just because you've got differing fraction or volume fraction of water. So if that term wasn't there, and you say you have a channel shaped like that, if it's static, common sense will say there's no flow in it, but if you didn't have that term in, it will generate a flow, which of course is nonsense. But it's something that is missed in models of this type. So that basically explains this term. Right, okay, so the key bit. Now where does the interaction between the forces? So Pi, which is the pressure in the solid phase, we assume it's related to the water pressure by this term here. So it's PW plus sigma. And basically what this term says is that if the fraction of cells is smaller than some threshold value, we're saying that there's no kind of stress being exerted. The cells are happy to be in that situation. However, if it's greater than this threshold value, then you're going to have a response which looks, so cells are happy here, is going to, I can't remember what the parameters, I think that was 5 big M. So there's going to be a fraction of space which the solid parts, I call it solid parts, but cells and EPS occupy, that if they're too densely packed, they don't like it, and they're going to push out. And that's basically what that term describes. So So that's basically model. So hopefully you got a sort of the idea of the sort of kind of the ideas between mixture fluids. I think it comes from soil mechanics and stuff. So so we had to solve this numerically for obvious reasons. Now at the time I became aware of level set methods to describe moving boundaries, which I was quite excited about because I'm not, well, I do a bit of numerical methods, but certainly not an expert, and I do like using finite differences. So when the level set became apparent to me, which works brilliantly in finite differences, I thought, yes, that's the way to go. So we decided that do a level set method. So basically, we have a variable, we introduce an extra variable, this gamma, and this gamma is defined so that it's 0 on the boundary. And we solve this transport equation, and this gamma equals 0 is going to move with the local velocity. So as the biofilm evolves, where gamma equals 0 is going to follow the edge of the biofilm, and that defines where the biofilm is. So it's quite a neat way to track moving boundaries. And to make things nice, normally, initially you solve you let gamma being the solution of the iconal equation, which is a distance function. So the distance function to the boundary is 0, but as you move away, it goes from 1 to... And it means that there's a small grade, well, an order 1 gradient in gamma, which means that the numerical method is quite stable and not so problematic. So we have this level set method. This is a scenario, so we just have a block of cells, same initial conditions before. On the boundaries, we assume that there's no 0 stress, so it's stress-free, and set the same boundary conditions and assume we've got some background pressure. Right, okay, so here's some numerical solutions that Mark Newstead did. So, so this is the evolution of the biofilm. Hopefully you can see it. So that's the initial conditions, and it sort of inflates out a bit like a ball. Because we've got 0 slip boundaries, there's a bit of a slow movement there. That's because we've got no slip boundary conditions there. And what we did a sort of kind of a local slip condition, which enabled things to spread out, so things don't get too singular. The reasons that has been outlined here at the point of contact lines, you get singularities. So, and here is the live cell evolution. So we just got your block and your inflating ball. It looks very flat on top. I think that's just a rendering of the graphics. But you get this sort of ball shape. In fact, it all seems to grow out in the same direction. And in the middle, you get your dead cells. So these cells are starved, so they die. And that's exactly the kind of thing that is observed in experiments. Comparing the 1D and 2D solutions, it shows that certainly if you're starting with a geometry like this, 1D is a fairly poor approximation of the 2D. Nutrients is coming in from all directions as opposed to only from the top, and you do get quite different results. It actually sort of grows faster in the 2D situation. So some other variables here. So he says, as I'm trying to remember what they are. So this is the water distribution. So so because the cells die, there's water in the middle, so you have sort of quite a fluid center part and sort of more of a porous medium outside. EPS, because we had a slight degradation of EPS, that again sort of reflects what the live cells are doing. Probably has the EPS decay rate a bit too far high, really. But it sort of shows you where the bits of the biofilm are likely to be strongest. So if you are going to exert a force, it sort of indicates where you might expect most resistance. And here's the live and death cells. So basically, as you expect, cells reproduce a lot on the edge, and that's the bacterial cell fraction. So
it looks like the EPS is preferentially selecting at the top where it's yellow. Is that just an artifact?
it could be an artifact of the thing. Yeah, because EPS is controlled by quantum sensing. I don't think I'll put the pictures in. So which is 0 on top. So yes, that would be an artifact. For some reason, I don't know if he sort of somehow lost the last couple of grid levels of the thing when he was doing the pictures. Okay, so the question, one of the original questions was, what are the kind of the flow regimes that you might get in such a setup? So if I point to the solid matter, so this is the live cells, basically everything's going out like a ball, and sort of the arrows indicated here seems to reflect that. The flow regime is quite interesting. This is purely a prediction. I have not seen any experiment that gives any indication of what the flow regime is like. But in this sort of setup, there are structures like this that do grow. They call them mushrooms, and it seems to indicate that you get a sort of I mean, these arrows look big, but there's a very, very small velocity. You get sort of a flow, most of the flow coming in from the top, and then a fair bit of flow going in the bottom. So if you're going to have streamlines, it looks like it's sort of doing things like that. So it's coming out the top and the bottom, coming in at the top and the bottom, moving out the sides. So as I said, I have not seen any experiments to say truth or otherwise of this, but that's what the model predicts. And this is just the sort of the stresses in the water, gives you an idea that, as you expect, that in the middle where there's no life cells, it's sort of pretty even, and it sort of increases as you go to the surface.
Are the flow fields on the same scale, the arrows? Because it looks as though the mass is moving out faster than the water. Outflow of the water, which would make sense.
I think it probably is.
Yeah, it doesn't matter.
Yeah.
That would explain.
Because it will be the same sort of scale. Everything's moving slowly, so it's yeah. And one of the questions that we wanted is something like this. So we have some idea of the flow regimes. If you're going to sort of try and understand what the motion of or the transport of some molecules, particularly large ones, where diffusion is a bit more difficult, so you want advection to draw in the parameters. So we just took a biofilm, assumed it was in a steady state, and we just so we added some sort of substrate that satisfies this equation here. So sort of there's the advection term, diffusion, and some sort of consumption. And these are the two pictures that sort of shows. This is a case where you've got the fluid flow. This is a case where you don't. So you can see there's a bit more penetration going on, but not a great deal. And we had to work hard in order to get pictures which look in any way different to the same. So we basically had to work with a molecule, assume a molecule which is about 100 kilodaltons, molecular mass of about 100,000, which is a massive molecule. So so you really do need big molecules to show any kind of difference in terms of the penetration, which is perhaps a little bit disappointing. It sort of suggests that the flow within biofilms is useless as a means to exploit things. Right, okay, so just a few concluding remarks. I presented some models of the biofilm growth. As I say, I was motivation was more on the quorum sensing side, but obviously I had to work hard at the fluids as well. So as with all these models, there's a significant reduction in the complexity. But a lot of the features that we predict, or the simulations we get, do broadly agree with what is observed in experiment. Unfortunately, experiments are not particularly sophisticated when it comes to analysing biofilms, but what there is, we have some at least qualitative match, but in some ways that's not too surprising. But we also did some other work, I haven't sort of shown it. So we made predictions regarding, okay, so the quorum sensing activity, which I did show, but things like, things like how you might sort of best administer anti-quorum sensing drugs or antibiotic treatments in a biofilm situation to sort of work out sort of optimal protocols. Well, not in any sophisticated way, but made various other predictions. So the core of the models, at least the fluid side of things, are essentially classical, Navier-Stokes, thin film, limits, Darcy's law, and the Brinkman bit. So all of that has been talked about here, and we've seen. The things that are slightly unusual in terms of the classical things is that the growth and death create a non-zero velocity divergence. You've got to change the states from cells to water, and that tends to give you a non-zero convergence. So you have to consider extra terms in the stress tensor when you're doing the appropriate thing. And also, it's a sort of thing in biology, in terms of energy, in fluids, you sort of think of heat as perhaps a sort of source of energy, or something like that, or just some force moving some part of it. Of course, nutrients is a means of energy input, so nutrients make things happen in biological systems. Yeah, I just want to point out, because this is a fluid dynamics thing, and just sort of kind of the challenges when you are dealing with biological problems is that parameters is very much an issue. And there's sort of things like reaction kinetics, which you can't avoid, threshold, turnover rates, diffusion coefficient, all sorts of 100 things, and even the data that you might be able to get is not necessarily reliable. And I put this in red because I know this to be true, that if you do see biological data with small error bars, they're lying. It's almost impossible. Even doing the simplest batch culture experiment, putting bacteria in a flask, trying to calculate what their population is every hour, you do 5 repeats, exactly the same circumstances, and you can get an order of 100 magnitude difference by time equals 12 hours. So repeatability is an issue, and as I say, I don't trust small error bars. And also in biology, that you don't get lots of nice physical laws like ideal gas laws and adiabatic formulas to reduce your systems. These things don't exist, unfortunately, and we have to sort of work with what we got. But lots of people in my field do love this sort of thing. I mean, I do sort of the small parameter stuff quite a bit, as you saw with the singular perturbation analysis. But there's a lot of people who do sort of parameter sensitivity, which in some way gives you lots of insight if it's done properly into models. So there are ways and means of coping with this as a backdrop of something in fluids. And just a final slide. So it's just basically sort of the role of where complex fluids feature in the life sciences. A lot of things that, if you haven't seen a talk like this before, you've probably been almost aware of most of the things in the first thing. Of course, if there's a flow going on, then yes, in some sort of biological context, then that's relevant. Blood flow, breathing in, tears, guts might be one you haven't thought about. It's a very, very, very complex flow problem. But there's things, these sort of side issues like microfluidics, flagellar motion, and motion of pseudopodia, where people recently have been using thin film equations to describe cell motion. But also porous media comes out a lot, because tissues, in a sense, are just basically a porous media if you look at it in a certain way. And lots of people using homogenization, things like tissue engineering problems, placental flow, highly vascularized sort of part, and things like vascularized tumors. And perhaps, as I say, if you haven't seen anything like this before, you might not have thought about a dense colony growing and expanding is in fact a very similar fluid problem like I just demonstrated biofilms. But it's the same in tumor growth, which I've worked in, and in tissue engineering, which I'm working in as well. So, okay, so hopefully you've got a sort of some idea of how fluids feature in a biological situation, and how we got to sort of think about other issues as well as the physics of the fluid when we're dealing with these things. So thank you very much for listening, and thank you very much for inviting me to speak.
Thanks very much. Let me just ask a question first. When you had the 4 components, the EPS, you didn't assign that a viscoelasticity or anything more complex like that, or did you, and I missed it?
Yes, we did. Yeah, so it's sort of part of this. Yeah.
But if the stress contribution to it is a viscosity down there, there's no elastic modulus.
Right. Okay. Yeah, you're sort of leading on to a very sensible question. Well, you are making a sensible question. Sorry. The kind of thing I've got sort of envisioned with the EPS is like if you take a ball of hair, so that we've got these strands and you poke it a little bit. The kind of resistance, there will be some sort of elastic response, especially if it's quite a large deformation. But if it's a little bit, you can move a hair over there; it's going to stay there. Very sort of small stresses, sort of envisaged that it can be described by a viscous fluid. It's very much an approximation. As soon as you put in any serious stresses, like even putting it in a slight flow, it is a viscoelastic substance, and yes, the modelling will have to be modified for that. Yep.
Question? Yeah.
All right. So you didn't put any active kind of stresses that you would imagine that is there, and also effects of wettability or something like that. For
With, sorry, with?
I mean, at least in myxobacteria, they need to secrete something in order to move. So even if they are not proliferating, I mean, even if they are not moving actively, it's just proliferation. They need to do that in order to move. And also, I mean, osmotic pressure.
Right. All these things are certainly important. So you're talking about myxobacteria, is that right?
Yeah. Well,
Yeah. So the modelling here, I've sort of more into Pseudomonas aeruginosa, who in different environmental circumstances do exactly what you describe. They have a means of spreading fast, but that's not what's classically called a biofilm, because even though it's a colony, a spatially expanding colony, they have flagella, so they are swimming. And they seem to be doing this on a petri dish plate. I've actually done experiments myself on this. So if the agar they're sitting on is a certain bracket of concentration, an intermediate concentration, they can spread very rapidly. And what they seem to do is draw water from the agar, which enables them to spread. And that process is in fact quorum sensing controlled as well via a substance called rhamnolipid, which is they call it a biosurfactant, which probably is a biosurfactant, but how it works is not entirely clear. But so that's a different type of colony and a different kind of environment, which gives I wouldn't call it a biofilm, but it is a colony. In terms of myxobacteria, I don't know the details of what they do as a means of spreading. It might be more the sort of the swarming behaviours, what I just described, might be more swarming behaviour, that, as I understand it. I don't know. Well, we could talk more about the details, but yeah.
Yes.
It seems like you've developed a sort of general aggregation model in some sense that you have them sort of like a blood clot. So how do you think this model would model a biofilm differently than it might model something like a blood clot, where you've got something aggregating from that?
So with blood clots, you have some sort of site, and then you've got these platelets and things just floating past. So and do the platelets grow and divide in the site, or is it just purely deposited? Right. Okay. So that's a slightly different mechanism. Yeah. I mean, it's not something.
You don't have the erosion that happens here.
Right. Oh, there's no erosion. As I say, there's very little. Yeah. I mean, most of the experiments on biofilms are actually done in the flow chamber, and things like bits breaking off is a well-known and frequently observed phenomenon. So yeah, I'm not modelling detachment at all. That's the next level of complexity, and I have to look at the viscoelastic limit as well in order to even approach doing that properly.
In those 2D simulations you presented at the end, what were you doing at your free surface? Did you have an equivalent surface tension?
No, it's just stress-free.
It's just stress-free.
Yeah.
On all, so the effective sum combined stress is stress-free, or is it just the solid bit, or just?
Right. No. Sorry. Yes. Yeah, stress-free on the solid bit and
Okay.
PW imposed. Yeah. Yeah.
The first part, you had sensitive dependencies on the precursor thickness.
Yeah.
Would you, and then you suggest, so that means something's missing physically.
The
Or is it okay to have a dependence on a small product? Sometimes it does happen.
As a modeler, I hate it. It means there's something I don't know. Yeah, it's so when I talked about the very briefly on the similarity solution, where you arrive at this ODE and you've got that missing condition, which is normally to close it you need that contact angle. It's the same problem. So it's a travelling wave solution, but the dynamics here is dominating what's going on rather than dynamics here, which when I started this, I hoped it would be. But John King, being a world expert, he told me, right, of course it's, it's going to be dependent on epsilon, but yeah, it took me a while before I got to that kind of level.
If you added the first nonlinear term that you might expect to be important in that region, would that tame that singularity?
Right, maybe sort of something like Van der Waals or something like that. That kind of, you arrive at the same problem because there's going to be a parameter in the Van der Waals approximation, for example, which you don't know, and that's kind of so there'll be a dependence on the expansion on that. But I'd expect you to, you're going to get, you will get linear expansion. It's just that instead of this epsilon seeming to govern it, it'd be whatever the parameter is in there, so it's the same problem.
One last question. Okay, so let's thank John and all the students.